So I just want to jump right in the middle of the subject matter. But before I do this, with apologies to people who have already seen this at the M&M meeting, I'd like to mention that we are now, the Cryo-EM is now represented by a graphics novel. And I found this in Sapere, in Italy. So it's a graphic novel that a young artist has drawn. It's really amazing. These are all ink drawings, beautiful ink drawings. And right here, and I met the artist when I was traveling with David Duracier and Esther and my wife through Italy. Okay, so we have to be very proud that we now have a graphic novel to refer to. So I just wanted to very much emphasize this this fact that I denoted in the abstract, the ability of single particle cryo-EM to furnish hundreds of thousands of images of molecules fast frozen in solution means that even rarely cited conformations corresponding to high energy states are represented in the data set. And so what it means is that a continuum of states is experimentally accessible for visualization. And current practice of cryo-EM structural research using maximum likelihood methods does not take advantage of this opportunity. And so I'm talking now about several years, I think 7 years of collaboration with Abbas Urmas and Peter Schwander. Peter Schwander is here at the University of Wisconsin in Milwaukee. And we have developed an approach to map the continuum in a low-dimensional space by employing manifold embedding. And from the observed occupancies, the free energies of the molecule can be computed. And this is really a breakthrough, this phenomenal breakthrough that we, for the first time, have the ability to experimentally access the energy landscapes and draw a conclusion from that on the behavior of the molecule. So here I'm just showing the players. On the left-hand side is Abbas, Ali Dashti, who did most of the work, most of the software development work, Peter Schwander. And then on my side, Cao is right here, Suffragette and Evan. Now, Evan is a graduate student who did a phenomenal amount of work in developing a GUI, and I'm going to show you some shots of the GUI later on. And we have a number of publications, beginning with a PNAS publication in 2014, and then bioRxiv depositions. And so it all starts, and I just want to say that I leave the real nitty-gritty details to the presentation and poster of Peter Schwander later on. So I just wanted to essentially tell you what we have been able to do with this. So it all starts with the idea of representing images as vectors in n-dimensional space. And the image, for the representation to make sense, the images need to be aligned. And that concept we already used in 1981 is the publication with Marien van Heel, who is also here. And so we essentially have point clouds and manifolds in the high-dimensional space. So aligned images, if they are similar, form clusters in the n-dimensional space, and similarity translate into proximity in the n-dimensional space. And continuous changes in conformation of the molecule produce an arrangement of vectors in a continuous manifold. So, but we are really not interested how this manifold, what kind of shape it has in the n-dimensional space. We're only interested in how the data are distributed across the manifold. So you have to essentially do something like a flattening of the manifold. We have to follow the manifold and produce some kind of a local coordinate system that curves around with the distribution of the manifold. And that's essentially indicated here in this slide. So instead of talking about 100-dimensional, 10,000-dimensional space and so forth, we're talking about a subspace that is at most 5 or 6-dimensional, where all the necessary information about the way the states are distributed, the states themselves are represented. So we should aim to preserve the geodesic distance, which means the distance on that manifold, rather than the Euclidean distance in this high-dimensional space. So in order to describe this whole process in a nutshell, I have to put this flow diagram here, which is essentially the flow diagram that was already presented in the PNAS paper. We determine snapshots orientations. Now, in order to make the process simple, we simply take the orientation assignments that are already made by the existing software. That is something that is not essential, because it can all be, everything could be done in the manifold embedding approach, but it turns out to be much more cumbersome to do it all together. So it was somehow much easier to split it into these two different processes. Okay, so we essentially deal with star files that come from Relion, and then this take simply the orientation assignments for granted. And then so we are talking about orientation aperture, and I'm going to talk about PDs, which are projection, which are projection sets. And so within each of the projection directions, we find a conformational manifold. So within, as observed from any projection direction, we are seeing an aspect of the particle as represented by the manifold. So then we determine the number of the degrees of freedom. We map to the space of the known eigenfunctions, select the subspace, and compute the energy landscape. So we can really do this. We can compute energy landscapes for each projection direction. And we can compile a 2D movie along the selected trajectory in that space. And we can repeat it for many orientations. But now comes the sort of the killer. We need to combine all that information across the entire angular space. So I'm calling here patch information from different orientations. And we talk about a propagation that we have to start somewhere. And then we can compile 3D movies along selected trajectories. So in between, we now have a representation that is a consolidated representation from all, that draws from all orientations. And then we interpret, and then we're finished. Okay, so, and as a reminder, transforming occupancy, this is simply the number of siting in each state into free energy map. I'm referring to RC1, RC2. We simply call them reaction coordinates because these are somehow parameters which we can use, and they're actually the eigenvectors that we obtain. So by taking a negative logarithm of the occupancy, we get a free energy landscape via a Boltzmann relationship, which means that a high occupancy means low free energy, low occupancy, high free energy. high free energy means that the states are difficult to reach at biologically relevant temperatures. Okay. So the first application, which we published in 2014, was with yeast ribosomes. We had yeast ribosomes, 850,000 images from cell extract. This was a rather rough kind of extraction and purification procedure, which means that no ligands were remaining. There were no EFG, EFTU, tRNAs, mRNA. So these are naked ribosomes. And at the time, we didn't have the capability to combine all views. So we just went around the equator, and that reduced the data set to maybe something like 100,000. And here are the eigenvalue spectra just to show you a selection for different projection directions. And you can see very clearly that one stands out very, very strongly, and it is in fact something that we already expect from the behavior of the ribosome in a thermal bath. It's the well-known intersubunit rotation. So the ribosome, as we know from previous experiments, rotates freely and through an angle of almost 10 degrees. And that's, we can see here, this is an example of a projection direction. And it uses the 3D projection view in which we can very clearly see the small subunit on top of the large subunit. And when we go along the first eigenvector, you can see the formation of this boundary there. And that means that along this boundary, the small subunit moves. So it's a clear indication of intersubunit motion. Okay, so by consolidating all this information, we were able to get this energy landscape. So it's factor 1 against factor 2. In blue is the trajectory of low energy. And just now in a landscape like this, we can simply grab individual states or a whole bunch of states, and then trace the data back to the various projection directions. So we can now form a 3D representation. So we can essentially do a 3D movie along any trajectory that we select. So we we selected this blue trajectory and looked at what kind of 3D structures are behind it. And lo and behold, we see conformational changes that we already know from previous studies that are associated with the elongation cycle. They are the intersubunit motions and the motion of the small subunit head. And we can recognize the different states that are represented. So we have this amazing fact that the structure, the ribosome structure in the thermal bath visits all the different conformations that are associated with the elongation cycle, even in the absence of ligands. And it's again a reminder that evolution really works not just on structure, but also on dynamics. So the evolution of molecules occurred in such a way that the architecture lends itself to optimum use of the energy that is around there. So I'm going back to the place where I encircled one box in the flow diagram, which had to do with the propagation of information across the angular space. So here in orange are the projection direction that have more than 100 particles. So we have to produce some kind of a threshold, because if we have less than a certain number of particles, the results get very sketchy. So this leaves this part, okay? And then we select what we call an anchor PD. And so this is the anchor node 90. And this is what we see. Okay, now, so in detail, if we now have these eigenvectors or reaction coordinates, this is number 1, and then this number 2, and so forth. Now, the first thing that happens is, if we now migrate to the next one, to a neighbor, it can happen that since this expansion is done completely independent from the other one, that we get the same information but with opposite sense. So then we have to make a correction. We say, no, it's the opposite sense. Okay, so we have to flip it around. That is one complication. We have to trace the senses of the forward or backward across. The other complication that can happen is that the eigenvectors are very close together, or the eigenvalues are very close together, which means that all of a sudden what was eigenvector 1 has become eigenvector 2, and then all of a sudden we have to sort of keep track of this. So this is a much more tricky part. Okay, so I just wanted to give you a sense here. Here are 4 neighbors of the anchor projection. You can already see some of them are the same sense, some are opposite sense. Okay, so we need to have some kind of unless we want to do that by hand, which can be extraordinarily difficult, we need to have some kind of an algorithm that does this. And so we are working on this. It is still not completely solved. We have success for tracking senses and so forth, but the other part of the flipping of importance of the different eigenvectors is more difficult. So it's an inference using belief propagation. There is such a thing, belief propagation. It's beautiful as a word, really. It's like rumors spreading out, and if the rumors all agree, then yes, this is the solution. So I'm just going to tell you about the Ryanodine receptor, which is a collaboration with Andy Marks and Wayne Hendrickson, and we have been able to get 3.6 angstrom resolution. And then we have gotten in the paper in Cell, we have various classes that show the receptor even in different states, in open and closed states. So this is really what's in the paper, but we have this entire data set available. They are the Ryanodine receptor without ligands, and the phenotype is that the channel is closed when we simply look at the class reconstruction. This is around 400,000 snapshot. And then another data set is with various ligands, with calcium ions, ATP, caffeine. And then so the phenotype is that we see the channel open. That's another 400,000 snapshot. And so these data sets were pooled into 800,000 particles to investigate all the changes on the same footing. So this is what we find here. We find now we represent this in 2 different energy landscapes, the ones without ligands and the other ones with ligands. So we can see that now to understand this, you have to see that for any point, whoops, I don't know where my cursor is. Okay. So for any point up here, you get to an equivalent point here. It's the same conformation. Okay. So you can immediately see that if you go at the minimum, the energy minimum at the start, and go down to an equivalent point, you see that there are no particles. And similarly, if you go to the finish point and go up there, you have no occupancy. So in order to get from one place to the other, the molecules really need to go uphill. So a very small portion of molecules get to this area of what we call a hotspot, and then they are able to then transit, and then they go downhill to the finish point. So in other words, the fact that the that the receptor works at all is due to the thermal fluctuations. It would not happen otherwise. And in order to find the hotspot, we can look at the transition probability, which is essentially the product of the two distributions, and that has a very high value in this area. So this is the absolutely highest one, but there are areas around it. So everywhere there's the ability of the molecules to transit. And here's just a straight shot of this. So we can sort of summarize this and at the same time display a structure. Okay, you can see that this inferred pathway is then associated with the opening and closing and activation of this channel. And it's of course it's of great interest to see how this compares with the maximum likelihood findings, and the classes are really all over the place. Some of them are really very nicely in the class 2, are very nicely in the energy minimum. Others are scattered and off and so forth. So essentially, we interpret it as saying that we are now, I mean, by projecting the findings of the maximum likelihood into this space, we might be in a sort of in an oblique position. We don't really know with the meaning of this of the selection. So I mean, it's obvious in a continuum. If you have a continuum of states, and then we use maximum likelihood methods, you can really from a slight increase in distribution density, you could just get a cluster, a spurious cluster, and so this situation happens if you really don't have very strongly defined clusters. And so what it means in terms of the atomic structure, we can now using this just showed you before how we can retrieve the structures in particular places in the landscape. So we can now compare the trajectory. First of all, we can make atomic structures, since we get into the range of 3 to 4 angstroms resolution in these reconstructions. So we can now have atomic models along the trajectory. And so what I showed you before can then be translated into 3D structures. And here we see the functional trajectory displayed, in these surface representations. And this here is compared with the linear interpolation. Very often people do morphings, and this is a morph between the start and the finish point. Obviously, it goes through a trajectory that has no experimental backing and comes to a completely different conclusion. Of course, nobody believes in results of morphing. Nevertheless, morphing is very often used to show how the structures, how the structural transitions occur. And I mean, the lessons from this here is really don't use morphing at all. It doesn't have any meaning because there's no experimental support in between. You only have the beginning structure and the end structure. So here, this is shown here by looking at the distance between certain groups in the certain atomic groups in the structure. In one case, it follows this what we call functional trajectory, and in the other case, it goes completely outside of what we find here. So next, I just wanted to show you some results with the ATP synthase. And this is really work in progress in collaboration with John Rubinstein in Toronto. And they have fantastic structures already available. And what we really want to know is how the conformational, how it actually works as a machine. And there are really beautiful ideas already on how it is going to work. So here are sort of snapshots of our GUI, and I'm going to show you a little movie later on. But just as sort of as a warm up to this, we have a representation of the structure, and then the projection directions around it, and then we have a way to highlight or select anchor projection. And for any selected projection, which is always displayed in the middle, we can then immediately view the corresponding eigenvectors. And then from there, we can obtain two-dimensional movies. So here are examples. Oh, I thought these were movies. Okay. All right, here you see an example. Here is a direction that shows you this kind of tilting of the C-ring. Here, this kind of tilting of the C-ring is very pronounced, and associated changes. Okay, here's another one. Okay. Okay, so from the way this whole analysis works is that the time scale, what we call tau, is exactly synchronized. There's a way of synchronizing these, which makes use of the number of sightings along the time scale. And so there is a synchronization that takes place. So we don't have to worry. So when I show you a movie, this shows you at any time, it shows you exactly the equivalent to a movie from a different direction. Okay, so here's an sort of a mock use of the of the GUI. We can select a direction and we immediately find the angular orientation. We can make a selection here how many minimum and maximum you want to have. And here's the same in the angular space. This is the distribution of PD particle occupancies. Set the number of eigenfunctions. Set an anchor. It's going to blink, okay? And now we can view this. You can sort of go back and forth with the slider. You can see the changes. Here's a manifold, and there is an editing tool. We can say, well, we want to get rid of outliers. And then you can see what the outliers actually show. Now we have removed the ones that we didn't want. And here we have a representation of the manifold in a three-dimensional space. We select among the vectors that we want to show. Okay, now this is a navigation that selects all the PDs that we want to contribute to the consolidated representation. It's an eigenvalue. You can see that the 5 first ones sort of stood out a little bit above the noise. Okay, so this is now a comparison of movies from adjacent projection direction in order to help you decide whether the algorithm has made the right choice of sense. Okay. And compile results across all PDs, and then from that we get an occupancy map. This has an error here on the side. There's some kind of an artifact, so we have to avoid this area here. Now, using the Boltzmann formula, this now has been translated into a 1D free energy path. The tool that we have set up right now, which has been in beta testing, in alpha testing, is only giving us the one-dimensional energy path. We decided to do this because we didn't want to start in a too complicated way. But the real aim is to present the 2D information that we had before. Okay, so I just show you some 3D representations for selected views of the structure. You can see what is going on here. These are very complex movements. So one gets a sense when one gets forward and backwards with the slider repeated times, you can see what kind of movements. There's an interior rotor which is sort of an interior rotor that one sees from different directions. Okay. So in conclusion, the analysis of large ensembles of molecular images captured by Cryo-EM reveals that molecular structure is not fixed, but occurs in multiple states in a continuum. And the free energy landscapes of molecules and molecular machines can be obtained by the manifold embedding approach. It is possible now to infer possible functional pathways from Cryo-EM snapshots that show the molecule freely equilibrating. So an outlook, far from settled and complete, single-particle Cryo-EM is a dynamic field which will have many surprises in the future. And I didn't talk about time-resolved Cryo-EM. This is another aspect of development, future development. It can give detailed kinetic information about the reaction of two-component system on a time scale of tens to 100 of milliseconds. The ability to retrieve multiple conformations is particularly forte of single-particle reconstruction, as it promises to reveal the functional dynamics of a molecule. So with that, I'll just show you the group last year on the Black Building. Thanks a lot for your attention.